So the next part of our innate immune system is going to be that inflammatory response, or it's just called inflammation. And so this is a response that's going to be localized, and it's going to be in response to microbes or to tissue damage. Things that are going to trigger or initiate inflammation, like the tissue damage. We're going to connect this to the complement system. I know I keep talking about it, but complement, it got its name because it complements all the other things of the immune system, or particularly 3 things. The mediators of inflammation are histamine, released by mast cells in the tissues or basophil cells. The cardinal signs of inflammation, and yes, you have to know this: redness, heat, pain, swelling, and sometimes a loss of function. We don't like inflammation, but there's actually a good purpose to it. It will prevent the spread of infection. It calls in white blood cells to dispose of cellular debris. It releases chemicals and alerts the adaptive immune system, and it also sets the stage for repair. Let's go ahead and watch. I know it's kind of like YouTube Day, but there's a lot of little moving parts to this, and I can talk about cells moving and release the chemicals, but I think it just makes more sense to watch it.
Crack. Yeah. Got start jump. Another one. Oh, dirty. Yeah. Come on.
Some more good music.
Inflammation is the protective reaction of vascularized tissue to local injury. Acute inflammation is the early, almost immediate reaction of tissue to the injury, and it is the first phase of wound healing. Acute inflammation can be triggered by cell or tissue damage, or by the presence of dead cells or noxious agents such as bacteria. Acute inflammation typically occurs before the immune response becomes established and aims primarily at removing the injury-causing agent and limiting the extent of tissue damage. Acute inflammation occurs in 2 overlapping stages: vascular and cellular. In the vascular stage, arterioles and venules near the site of injury constrict briefly, then dilate. Dilation promotes congestion, while an accompanying increase in capillary permeability leads to the movement of fluid into the affected tissue, resulting in the 5 classic signs of inflammation: redness, swelling, heat, pain, and loss of function. As fluid leaves the capillaries, the blood remaining in circulation becomes more viscous, it flows more slowly, and clotting occurs. The cellular stage of acute inflammation is initiated by the movement of phagocytic white blood cells, or leukocytes, into the area of injury. The leukocytes begin to adhere to the vessel wall, and then, in a process called emigration, squeeze through the wall and move into the inflamed tissue. The leukocytes wander through the tissue, guided by chemical signals in a process called chemotaxis. The cellular stage culminates in the leukocytes engulfing and degrading the bacteria and cellular debris in a process called phagocytosis. Products of phagocytosis, along with plasma and blood cells, form exudates, which accumulate, causing swelling and pain. Exudates are composed of serous fluid, red blood cells, fibrinogen, or tissue debris, and white blood cell breakdown products. Concurrent with the events of the vascular and cellular stages, chemical mediators release bioactive agents that act to mediate the inflammatory response. Mediators are derived from the cells or from plasma. One of the first mediators of an inflammatory response is a cell-derived mediator, histamine, found in high concentrations in the mast cells of connective tissues adjacent to blood vessels, as well as in blood basophils and platelets. Histamine is released in response to a variety of stimuli and causes dilation and increased permeability of capillaries. Serotonin, another cell-derived mediator, performs similar actions. The 3 major plasma-derived mediators are present in the plasma in precursor forms that must be activated, usually by a series of proteolytic enzymes. The kinins increase capillary permeability and stimulate pain receptors. The clotting system traps exudates, microorganisms, and foreign bodies. The complement cascade causes vasodilation, promotes leukocyte chemotaxis, and augments phagocytosis. Acute inflammation comprises a variety of complex protective responses to injury that together lay the groundwork for the next stages of the body's recovery: immune response and tissue repair.
I just love it when people add some jazzy music to those things. All right, so I gave you a different picture in your notes, but this basically says the same thing, a little more pictorial. So we have either trauma or it can be in response to microbes. Either can kick off inflammation. Main mediator is going to be histamine, but there are those other inflammatory mediators. The big thing that's going to happen is capillaries are going to get leaky. And so we're going to get that diapedesis. We've got some chemoattractants are going to call those white blood cells into that area. Then we have our phagocytic cells are going to destroy and remove the invaders, cellular debris, and then once again we sort of see that as pus. And I don't know if you caught that on that first episode, we were talking about tonsils, but we saw those palatine tonsils, they were inflamed. You saw how swollen they were. So redness, heat, pain, swelling. You even saw pus in those tonsillar crypts. And so the inflammatory response, we don't like it, but it has a good function. It's going to prevent the spread of damaging agents or pathogens. It's going to dispose of cellular debris. It's going to alert the adaptive immune response and set the stage for repair. That's just some verbiage for you if you didn't get it from that video. So let's look at this. So it shows tissue injury. And like I said, those inflammatory chemicals, there are others. The big one I want you to know is histamine, but we're going to complement the kinins and prostaglandins. And so we get leakage. So arterioles dilate, increase capillary permeability. And so we're going to get neutrophils, monocytes, and lymphocytes drawn to that area. That blood, because those arterioles are going to dilate, we get more blood flowing to that area. So local hyperemia. So we have more blood to an area. So that's why it feels warm, because wherever blood goes, it's warm, and you get redness. The increased capillary permeability, when the capillaries get leaky, that's when we get excess fluid in the interstitium. And so we get that swelling. And then too, a lot of those mediators of inflammation also signal pain. If there's enough swelling, you can get temporary loss of function, but you've all had swelling and you can still use your fingers, so you get the idea. What's leaking out too, we get those clotting proteins that help wall off that area of an injury. So it's a temporary fibrin patch, and then also that scaffolding for repair. So the whole idea is we bring about healing. We've brought in the white blood cells. Other things, inflammation, we get release of leukocytosis inducing factor. So we're going to get leukocytosis, because I told you that white blood cells, that count is going to be regulated. We've got maintenance numbers, but also when there's an infection, that count goes up. We get those leukocytes going to the area. Margination is when they stick to those walls of those vessels. Diapedesis, they squeeze through. And then we get phagocytosis, so the area is cleared. So hopefully that kind of helped put that all together for you. So redness, heat, pain, swelling, sometimes loss of function. And the idea is it will remove an invader, prevent spread, and set the stage for healing. So acute inflammation is short-term inflammatory response to an insult to the body. This is a good thing. Like I said, we don't like it because it causes pain. If that cause is not removed, so it's not resolved, that can lead to chronic inflammation. So I'm trying to say acute inflammation is good, it has a purpose. Chronic inflammation is bad. It is that ongoing inflammatory response, which there's another textbook kind of talks about inflammation. Think about a sprinkler system in a building. So if there's smoke somewhere, we get the sprinklers turn on. So there's damage because of that water damage to the sprinklers. The idea of it was good, but if you have too much inflammation, what you end up with is you get tissue destruction. And so an example of acute inflammation would be like rheumatoid arthritis. And so that's due to autoimmunity, and that chronic inflammation damages those joints. So that's just an example. So you get tissue destruction, and instead of regeneration, which is what you get up here with acute inflammation, you get fibrosis, you get damage. So acute inflammation, good. Chronic inflammation, bad. So now we're going to talk about some antimicrobial proteins. And that first group we're going to talk about are the interferons. So interferons are made by virus-infected cells. So when a cell gets infected with a virus, it releases interferon. Remember, viruses can only reproduce inside a cell. Then typically the virions are released from that cell and then they go to infect the neighbor. So this is just a warning from that virus-infected cell. That one's a goner to the neighboring cell. So kind of this little run you fools. What it does is that it causes these neighboring cells to basically make an antiviral protein. So if this cell gets infected by that virus, it is going to go through apoptosis. So it's basically going to kill itself to stop that chain of infection. So it prevents the adjacent cells from becoming virally infected. Other interferons, other things, it also stimulates phagocytosis and it helps resist infections, including tumor growth, but it's in response to viral infection. So finally, we're talking about complement. So complement gets its name because it complements other branches of the immune system. And so complement, it's going to be a cascade, so similar to the blood clotting cascade. The majority of these proteins are going to be made by the liver and they're made in an inactive form. And so there's going to be this cascade. Don't get caught up in the cascade, but I really want you to watch and see the effect of complement. So I'm not going to ask you about C1, C2, C3. Once again, sadly, they were named in the order they were discovered, not in the order that they function. But let's just go ahead and watch.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5A and C5B. C5A enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Okay, so there's different ways that complement can get stimulated. So the classical pathway is through antibodies, and that's one of the ways antibodies can neutralize things and target them for destruction. The less than pathway, alternative pathway, these are just going to be in response to things that bacteria have, that fungi have that we don't have, so components of their cell wall. All pathways lead to C3, and the three things that I want you to know that complement does is it leads to opsonization, and we talked about that's that coating of a pathogen to enhance phagocytosis. C5a is going to enhance inflammation, so it stimulates histamine release, so we get increased blood vessel permeability, we're going to get phagocytes, and C5 is going to act as a chemoattractant, so chemotaxis, so call those white blood cells there to then engulf and destroy. C3b, 5, 6, 7, 8, 9 makes something unique called the membrane attack complex. I just remembered it Mac attack. You're going to think I have this obsession with Big Macs because I use Big Macs to remember something else too. But that actually inserts a hole in target cell membranes. It's really good for fighting against gram-negative bacteria. If there's a hole in the membrane, then that means they lose the integrity of the membrane, and it's going to lead to cell lysis. So you get cell lysis from complement. The last part of our discussion on the innate immune system is fever. So fever is a systemic response initiated by chemicals, particularly IL-1 and IL-6, so interleukins, but they're called pyrogens. So if I'm a pyromaniac, I love fire, so the genesis of fire. What these chemicals do is they reset the thermostat, which is in the hypothalamus of our brain. So normally 98.6 or 37 degrees centigrade is our set point. What happens, our thermostat gets turned up, so fever is 100 degrees Fahrenheit or higher. It is definitely an indicator of infection, particularly a bacterial infection. So if someone has a fever, they have an infection. What fever does is it's going to mobilize the body's defenses. It excites those phagocytic cells, makes them more attackative. Also, it causes iron and zinc to be sequestered by the liver, which inhibits pathogen growth. These are often limiting growth factors for bacteria. And so it's going to speed the repair process. But the good thing, if someone has a fever, it is an indication that they have
disease deep inside me, makes me feel uneasy, baby. I can't live without you. Tell me, what am I supposed to do about it? Keep your distance from it. Don't pay no attention to me. I got a disease
and a fever.
I got a disease.
Okay, so we have discussed our first line of defense, which are surface barriers, so skin and mucous membranes. And then we've just discussed our second line of defense, so those phagocytic cells, so neutrophils, macrophages, dendritic cells, our antimicrobial proteins like interferon and complement, the inflammation, our inflammatory process, which is going to prevent spread, call the immune system and alert them, and then set the stage for repair, those natural killer cells that kill by cell-to-cell contact, and that systemic response to an infection, fever. So come back for our last part of the immune system, that third line of defense, the adaptive specific immune system. You guys have a great day. As always, if you have questions, feel free to ask.